Okay, good morning, good afternoon, everyone. Welcome to GOBii November web seminar. So as people are joining, I'm going to just share the ground rule. There's a chat function. Please help yourself with question, comments anytime by typing in your chat. And at the end, we will unmute everyone for questions. So today, I'd like to introduce you our host, Dr. Elizabeth Jones, GOBii Director, to introduce our speaker. Yeah, with that, Liz, please go ahead.
Thank you, Star. Hello to everyone, and thank you for joining the GOBii seminar series this month. It's my pleasure to be able to introduce Iain Milne. We've been working with Iain at the James Hutton Institute for the last 3 years, and they're a fantastic group to work with. Iain is head of research computing at the James Hutton Institute, and he provides HPC and Linux facility to over 200 members of the staff there across 2 sites and in areas of genomics, transcriptomics, and variomics of crop plants and their pathogens and pests. And not only does he do that, but he also leads a development group. They develop visualization tools and decision support tools, primarily Flapjack, and Flapjack is the tool that GOBii has been working with the James Hutton Institute on. And that's for genotype visualization and analysis. And he also works on Tablet, which is a visualization tool for next generation sequence assemblies and alignments. So welcome, Iain, and I'll hand over to you now. Thank you very much.
Thanks, Liz. Thanks, Dar, as well. And I guess thanks for giving me the opportunity to talk. So yeah, this is just a sort of high-level overview of some of the tools and visualization programs and things that we've developed over a good number of years here at the James Hutton Institute. One of my sort of job description roles would be research software engineer, which describes people who develop the software that the researchers use, but don't necessarily actually, I don't necessarily use the tools myself. So my background is all computer science rather than biology or things like reading. So it's an interesting topic for me to be in. So just briefly to begin, if the slides would actually move on, I've lost my cursor. So just a brief bit of background on the James Hutton Institute. So we were formed back in 2011 from a merger from what at the time was the Scottish Crop Research Institute, which is where I was based, or still technically based, and the Macaulay Land Use Research Institute, which is up in Aberdeen. And they came together to become the James Hutton Institute. And one of the sort of the questions we always get, because we didn't know at the time, was who James Hutton actually was. So I went through a long phase of just describing him as some old dead guy, but for the sake of this talk, I thought I'd actually find out properly who he is. So he's described as a leading figure of the Scottish Enlightenment, which was this 18th century golden age, apparently, of intellectual and scientific achievements. And he's regarded as one of the founders of modern geology and one of the first scientists to describe the Earth as a living system. And apparently his thinking on natural selection influenced Charles Darwin in developing his theory of evolution. So it sounds like he was quite a big guy, and he's given the name to the Institute now. And we cover, as it says, it's all sorts of research, obviously on crops, which is what we're primarily involved in, but this stuff on land use and soils and environmental research and things. So it's a good team that's been brought together across the 2 sites, and I think it's about 400 or 500 researchers over those 2 sites now. In terms of where we are, then this slightly stereotypical map of various countries, you probably all know where we are within the currently disunited kingdom. If ever a country was ever more split down the middle because of Brexit at the moment, the UK is probably it. And within the UK, we're in Scotland, which is in the upper half. And those of us doing the software development are based here in Dundee, which actually has quite an interesting history in terms of discovery and research. So the ship that took Scott to the Antarctic, the RSS Discovery, is actually based here now. And as you can see from the little graphics, we've had lots of involvement in video games and video game development from local companies. So things like Grand Theft Auto and Lemons were both developed here. So we're in Dundee, which you can see is about an hour north of Edinburgh. on the Chile northeast coast, and Aberdeen, which is where our other JHI site is, even further up the coast there. It's just a quick advert for Dundee itself, because it's a really, really nice place to live and work. Obviously, being in Scotland, the scenery is fantastic. We have easy access to skiing if you're into that. We are far enough north that we can quite often see the northern lights, the auroras. This time of year, we're far enough north that it's probably dark enough middle of the afternoon to see things like this. We have this fantastic new museum of arts and design that just opened this summer, and it's really increased the number of people coming to Dundee. So it's well worth a visit if you ever get the chance. So I'm in a group called ICS, Information and Computational Sciences, and we're sort of split across these three kind of zones.
I am, yeah. I can't hear Ian. Is that right for you too?
Right.
Must have just lost connection.
Okay, let's see.
Oh, it's muted. I never lost, but it dropped my screen share, and the same thing happened on Gordon's. Can you hear me?
Yes.
Yeah, we can hear you again.
Welcome
Okay.
back.
Look at your screen again.
Yeah, I mean, it didn't drop the actual connection weirdly. So where did I get to?
You started sharing now. It's good.
About the computational
So
department.
it was on this. It was on this slide.
slide. Yeah, we
That's
saw that
fine,
one.
yeah. Okay. So there's about 13 or 14 bioinformaticians split across. These aren't subgroups. It's just sort of areas we work in. So I'm heavily involved on the infrastructure, research, computing side of things, which relates to the HPC and Linux, and then also on the information systems side, where as it says here, we develop applications, databases, tools to facilitate the analysis, quality control, and visualization of genetic resources and plant breeding. And that's the kind of tools that we've been working on. That's what I'll be talking about for the rest of this talk. So I've lost focus yet again. So in terms of the people involved in that, we have this little informal subgroup called, well, software development group, and a Twitter handle of Crop Geeks. So there's myself and Gordon Stephen, who have most closely been working with GOBii. And then we've got some of the other developers that will work on, so the tools I will talk about are not all of mine. So we've got Sebastian and Paul that are heavily involved in tools like Germinate and Helium. Linda that works on some of the websites and the databases. And then David, who some of you may know, who up until very recently was the group leader for information and computational sciences. He's slowly winding down towards retirement now, but he's still around in an advisory role for us. So that's the team. Very quick overview just in terms of the kind of things that we've been developing. So it's a wide range of visualization, database analysis, and decision support tools. Some of them going back 10, 15 years now we've been working on this, covering areas, things like sequencing, genotyping, phylogeny, statistics. Probably primarily desktop-based tools. That's certainly the area I'm most comfortable with. One or two web-based tools. And more recently, some of the team we've been branching out into doing some mobile application development as well. Okay, so why do we do visualization? As it says here, the ultimate goal is to try and provide insight into complex biological data. And the reason visualization works is, and the reason it can help with exploring and understanding large amounts of information at once, is just because your eyes are really good at certain things, it says here, such as edge detection and pattern recognition and discerning differences in color. And it's based on this assumption that sort of visual representations and interaction techniques, we can do certain things, take advantage of your eye's ability to see and to explore and to understand large amounts of information at once. And again, that's actually easier if I sort of give you a sort of really simple visualization example for that. Some of this works over Zoom, is that if I throw up a slide full of numbers and I ask you to find the largest number, then obviously given enough time on a small slide like this, you'll be able to do that. But if I change that to something visual, then I would hope that almost straight away your eye should be drawn to the largest square there. And that's just something that happens subconsciously. Okay, so we have a range of tools, and the first one I want to talk about is called Germinate 3. As it says here, so it's a generic plant data management system designed to hold passport data and a range of additional data types. And I'll go into what that means in a moment. And the reason I want to start with this one is because it acts kind of like a hub from which all of the other analysis and visualization, well, not all of them, but from most of the other analysis and visualization tools that we have can connect into. Germinate is one of the web-based ones. It has a MySQL backend database. It's very efficient. It's been optimized for very large genotypic data sets, using methods very similar to what the GOBii database does. in terms of having HDF5 backends, but also has a very powerful and user-friendly front end to it as well, developed on the web. And a lot of the visualization stuff that we can do can actually just be done directly there. When we need to get into the sort of heavy lift inside of visualization, that's when we can move to the desktop tools. So yeah, so Germinate will store quite lots of different data types. So you've got passport data, pedigree data, obviously genotypic data, which is important to us when we're talking about tools like Flapjack, field try data, and so on. And the concept for Germinate was just there was lots of different types of data that projects gather over time. You need to make that data available, ideally through a single repository. And then if you've got all that data in one place, then it makes a lot of sense to be able to try and do at least as much of the browsing and the searching and visualization as you can in the same place. Germinate is really what we class as a data warehouse. So it's designed for finished data rather than working data. You probably look at the kind of stuff that Dart and that produce on that side of things. So we have lots and lots of different instances of Germinate for the different crops of both that we work on in-house here, so things like barley and potato and the soft fruits, and then the various work that we've done with collaborations around the world. So obviously Maize and Wheat with CIMMYT being one of the main ones. And the guys have just started on a major new project recently with the Crop Trust, which is going to have, I think, 13 or 14 different species all being put into Germinate and made available to the public and to the world at large. So just a few screenshots of what Germinate itself looks like in action. Regardless of the crop that's in it, there's a common interface designed for doing this. So this is the front page in terms of where you would start, and it can be customized to the project itself at this point. So this particular screenshot is from one of the crop wild relatives data sets. So this is on eggplant. I can't guarantee that all the screenshots will be on this one. You have views like this. So when we're listing the sessions, you just have tables that will bring the information back very quickly. You can sort them. You can start to select and group information and collect it in the concept of a shopping basket as you go along through Germinate. You can do lots of visualization. So we've got some scatter plots here on doing some comparative data. We have lots of information on maps and geographic information. So you can see where you have data stored around the world. You can overlay climate information on it. You can do selections of data over the maps as well. So you can click and drag and highlight areas and find out where the data is within that. So lots and lots of different things you could do with Germinate. But in terms of getting into the tools that I work more closely, like Flapjack, then it does actually have that screenshot in there. In terms of dragging things like that, because these were all embedded on a single slide. In terms of getting data into Flapjack, then we go through the genotypic extraction process with Germinate. So depending on where you stand, I think in this case we've switched to a different data set. This is the Seeds of Discovery project with CIMMYT. So this is some maize data. So you go in and you'll say that you're after some genotypic data. It would then show you, okay, well, these are the data sets that we have stored within Germinate. You would select a data set, at which point you would then say, okay, well, you know, which maps are you interested in, which marker groups, and so on. And then you have these options within Germinate itself that it can actually create the Flapjack project for you. And so long as Flapjack's installed, then you have a really simple option that says, okay, I just want to send that to Flapjack, which at that final step would actually open up that data into the Flapjack visualization tool. So that's the one that we actually want to cover next. So rather than talking about it on this screenshot, I'll jump forward and just give you a brief overview of Flapjack. As Liz was mentioning, so it's a tool for graphical genotype visualization. We would certainly hope that it's a high performance tool. It's easy to use. These tools are easy to install. And it started off historically as just a visualization tool, but over the years, we've added more and more analysis modules to it, and especially with the work that we've done with GOBii. More recently, that's where a lot of the focus has been on. So turning it into more of a decision support tool as well. So we've got various modules in there to do things like principal coordinates analysis, generation of dendrograms, and some of this stuff links out to some of the other tools that we have and actually links back to our compute cluster as well. So some of this analysis is actually running on HPC rather than just on the desktop. And then some of the statistical routines that we have developed with GOBii, so ones for doing marker-assisted backcrossing, pedigree verification of lines, and forward breeding as well, which is what we're working on, also in collaboration with the HTPG project as part of EIB as well. It's all just acronyms. So just a sort of brief overview of some of the various views that you get in Flapjack. I'm not going to go into too much detail here, but I'll talk about the main visualization in a moment. But the views here show various different styles of visualization and the color schemes that we have. We have Excel-style views when we're looking at some of the statistical results, and all of these views are all linked together as well. So when you change things in one view, it will reflect in another. So in terms of the display itself, this is a Barley data set, I think, that was loaded into Flapjack. And assuming you can see the mouse, then what we're dealing with here, and the main view, what you get is a list of the lines that you have is shown down the left-hand side here. And then we have a map across the top in marker order. And then the main bit of the display is this graphical genotype of the actual allele calls for that data. This is using it's using Flapjack's nucleotide color scheme at the moment, which is simple enough. Obviously, we just give a different color to the A, C, G, and T. Where the alleles are homozygous, we just score them once. So, we see like T, G, and A here. And then where the heterozygous, we actually do this little split diagonal. So we've got an AG here. We can bring in phenotypic information. So we actually have a heat map of phenotypes down the left-hand side here. And we can bring in overlay stuff as well. So we've got some QTL across the top as well. You have an overview down here on the left-hand side. So this is actually showing just one of the first chromosome of Barley here. And this little red rectangle shows where we are in the overall chromosome in terms of the number of lines that we have. And so this main display here has been reflected in this little red rectangle. So some of the things we can do with it, this is a data set that was a PopSeq sequence mapping population of Barley, where we actually have, you can see that there's more data for the first two lines. We've got two parental lines at the top here. And they were sequenced, and what we can do in this example is if we select all of the markers, so once they were sequenced, this obviously went into contigs at that point, and we can select all of the markers from a particular contig. So the marker names are actually based on the contig names in this particular case. So you can see the data looks fairly random at this point. So if we select all of the markers from that contig, and then we can actually order them by their similarity to one of those parental lines, and you can see we're starting to get a little bit more structure here. And we can go a wee bit further by actually bringing in some of other Flapjack's other color schemes and having the data colored up by their similarity to one of those parental lines. So you can see you have quite a nice red-green split here. So all the red alleles are coming from one parent, and all the green alleles are coming from another parent. And this is great for things like quality control. If we go back to them, you can see we have these blocks here where you've got a whole bunch of data that's potentially coming from one of the other parents. So in terms of looking for errors or looking for possible recombination breakpoints and things. And it shows up quite nicely down at the bottom here. This is a little graphic that's actually showing, so for wherever the mouse was at the time this screenshot was taken, all of the line under that mouse is represented here, regardless of the zoom level. So you can see you have a sort of nice block of data coming from parent 1, and then it switches to parent 2, and then some possible, are these recombination events, or are they just errors? And then another big block of probably definitely coming from the other parent at the end there. So you can do some nice things with it visually and just sort of see that with your eye rather than actually having to look at the raw data itself. As I was saying, we can do, if we go back to the other data, we can do some things like generation of similarity matrices, which in addition to the raw data itself, we can visualize that. And we can take that information, and this is one of the examples we have where we can throw that off to the HPC cluster and run some further analyses on that to get some dendrograms, which we can bring back into Flapjack. And once we have that into Flapjack, then the lines in Flapjack's visualization will reflect to show the sort order that the dendrogram has. And then one of the other types of analysis based off that same similarity matrix is to do some PCOA analysis. And that's where we bring into one of the other tools we have, which is called CurlyWurly. As it says here, so it's a multi-dimensional data viewer with a particular focus on the outputs of things like principal coordinates analysis and principal components analysis. And CurlyWurly it's a nice simple tool, but it's really, really clever. It's really, really fast. And it has a whole bunch of nice features that were added at the bequest of CIMMYT in terms of being able to deal with multiple categorization of data points. So, for instance, if you had lots of phenotypic information in Flapjack at the time you generate the dendrograms, then all of that information is actually passed through to CurlyWurly, and you can break down the data in quite nice ways based on that information. So CurlyWurly's view looks like this. So we have these categories of information down the left-hand side, and one of them will be selected based on how it's been coloured up. So again, this is barley data. And in this case, yes, so it's been coloured up based on seasonal type. So we have both spring and winter types of barley, and you can see genetically how they differ. Actually, probably not so much in the screenshot because of this globe thing, which I'll talk about in a moment. But if the globe wasn't there, we would have quite a sort of nice block of the winter types over here on the left, and a nice block of the spring types over there on the right. And this, what this is showing here is some nice abilities within CurlyWurly to select data. So you can pick any point in 3D space, so where each one of these little spheres represents one of the lines of data that you had back in Flapjack, say. And you can click one of these points, and then you can expand this exploding star out in 3D space, and it will grab points within the influence of that sphere as it grows. So it's just a sort of nice way of grabbing clusters that are actually close to each other in 3D. And then you can still go on to grab other outliers as well. So being in 3D, the easiest ways to try and show this, I have no idea how well that's going to work over the Zoom. I'm just looking back at Gordon's screen, which is connected to it. It looks like it's animating it not too bad. CurlyWurly copes quite comfortably with tens of thousands of lines, and the performance is actually really, really good for it. So this is, again, that spring and winter data, and you can see how the two clusters form quite nicely there. This is a few other examples of some of the nice data you can get with CurlyWurly. So that this is, again, I think this is wheat data from CIMMYT and the Seeds of Discovery project. I'm not going to go into any details on here, but you can get some really nice visualizations and some really nice clusters of data. And CurlyWurly is just a really nice way of playing around with it. And again, one of the things you can do in terms of the selections and what you, the choices you make in the tool, all of that information can be reflected back to applications like Germinate, in terms of how we actually link things together. Similarly with Flapjack as well, you can go and query information on the lines or the markers that you see within Flapjack, and Germinate can reflect that back for you. So we have a sort of very lightweight but very functional way for our tools to actually talk to each other, and I'll mention a bit more about that later on in the talk. So we have another one called Helium. So one of the ones developed by Paul as part of his PhD, which deals with pedigree visualization. So again, it's a nice little tool for facilitating readers and researchers to make more informed decisions related to the plant lines that they want to use in subsequent crosses. So I'll just go through this very briefly again. There's a sort of common theme to the way we develop our tools. We're very keen on being able to do overviews in addition to in-depth views of things. So you have something similar here, that there's a sort of overview window up on the left-hand side here. We have this graphical pedigree of probably yet again barley data in this case, and then more zoomed in views that can break down and actually show you the bits that you're interested in. Again, we can bring in the phenotypic information that's stored in Germinate and pass through to Flapjack and so on. So it's all linked together that way. Let's see, I think, yeah, on the next one. So again, the same sort of thing here. We can bring in the spring and the winter types, slightly different color scheme this time. So you can trace back the ancestry of the lines and see how they work that way. This is actually probably showing a better overview as well in terms of what we have in the main display being reflected here. Tablet is our tool for dealing with second generation and, well, I suppose even third generation sequence assemblies and alignments now. This is a tool that for a good number of years, probably around the time the James Hutton Institute was formed, we really spent a lot of time on this. It's been a very popular tool, probably the one with the highest number of user downloads and things. But it's probably fair to say it doesn't get quite so much love these days in terms of development time. It's a bit of a shame because it's a tool that we really enjoyed working on, but it probably doesn't fit into the plant breeding space quite as nicely as some of the other tools that we have that work better together as a suite of tools. And it's the same sort of idea. So this time, rather than obviously dealing with lines and markers and things, you're dealing with potentially tens of millions, if not hundreds of millions of short sequence reads broken down into various contigs, which again can even be up into the millions, which was interesting for us in terms of developing Tablet, because one of the problems we had with some of the visualization tools for dealing with NGS data was that they're very human focused, and that, you know, they assumed that you would only have a small number of chromosomes or a small handful of contigs, and you had very good reference data to work from, which is certainly not the case with what we have with plant organisms and some of the pests and pathogens that we deal with as well. So Tablet was optimized in certain ways to help people deal with that in terms of filtering their information down. And, you know, if you had a test assembly with, you know, millions of different contigs, and it was easier to work with that you didn't just have a combo box to try and find the contig that you were interested in. Similar to Flapjack, lots of different color schemes in terms of dealing with how we show nucleotide data, or how we show the different reads and things. So again, it's the same sort of idea. We have this overview window across the top here, showing this particular config, which has got, as it says here, about 5,500 reads in it. Yeah, again, the red rectangles show where we're zoomed in the main region. And we have these nice little tooltip overlays that give you more information on the particular read under the mouse. So this would be this red one here at this particular point. The shadowing effect is a nice thing we did in Tablet, where all the reads under that, for that particular nucleotide position under the mouse are then highlighted, so you can see where they sort of stack together that way. Information on the position of the reads, a little graphical overview of what the read looks like, and so on. Some histograms of coverage, annotations that can be brought in, so where it's marked up potential SNPs based on perhaps some GFF data that was brought in that way. Different color schemes, obviously, so the nucleotide one here, we've got ones based on read length and read types and so on. And you can see some probably bigger, either config or gene annotations being brought in this particular example. And you can do nice things as well in terms of highlighting where you've got the mismatches between the reads and the reference data, which is shown across the top here. So you can see in this particular case, you've got an example where there's lots and lots of errors towards the ends of the read. So that's probably issues with the machine itself, or just in terms of the Illumina technology that was used to deal with it. Or these ones here where we have massive numbers of mismatches to the reads, sorry, to the reference, where you probably are just dealing with read errors at that point. Other nice effects, you can really darken this down on the tablet itself. You can de-emphasize data where it is the same as the reference and just highlight where the mismatches are. As you can see in this case, where we've got these columns of mismatches, which are, again, are sort of probably really good candidate locations for SNP data, which show up really quite nicely when you do it graphically that way. Okay, so just a sort of final point on the applications themselves. As we're saying, we've been branching out a bit more into the mobile application side of things. So there's a few applications around it related to that, especially on the Germinate side of things in terms of data to help, in terms of tools to help get data into Germinate, in terms of scanning barcodes and scanning data and just field collection things. And then there's another nice little tool called Batata, which is actually the Scots Gaelic word for potato, which is used, developed in collaboration with St Andrews University and farmers in Kenya, I think it was, to just try and help identify crop diseases or diseases in potatoes specifically. So just nice little tools within that. I'll have a slide up at the end that just shows you where you can get more information on all of the stuff that we have and actually download links and things. Everything, I'll say just now as well, everything we do, we produce is open source and freely available. I've forgotten the slide was in here. Yes, this is just, as I was saying, something the tools that we have been very popular, which has been good for us. This is probably a map showing the distribution of users that we have for tablets. We can break this down in terms of the Flapjack and CurlyWurly and so on. It's just sort of nice to be able to visualize that for us as well in terms of where around the world people are using this. So as you can see from here, I mean, we've completely swamped Europe in terms of user downloads, as you perhaps expect. I mean, we're well on the way to conquering the United States, but for some reason nobody's interested in doing plant breeding up in Greenland, and we just can't work out why that might be. Okay, so a few other slides to finish off. This is based on some information or some stuff from the GOBI annual meeting over the summer. I thought it was just worth going through it again briefly here in terms of, so while we have these different tools, there is an integration challenge that we have in terms of bringing them all together, both within the suite of tools that we have and in terms of how we actually have to integrate with GOBI and the other project partners that we work with. And there was various examples related to that in terms of, so the server to client scenario, where we've got things like the GOBI extractor tool that has to send genotypic information to Flapjack, and the reverse, we have client to server, so for instance, CurlyWurly sending information back to Germinate, server to server. So we have those analysis modules and Flapjack, for instance, are wrapped up in Galaxy as well. So you don't have to launch Flapjack. You could actually do them in Galaxy. And we have scenarios there as well where you've got, Galaxy has to then get that data from systems like GOBii or from Germinate. And then the client-to-client ones on the desktop as well, in terms of being able to take information in tools like CurlyWurly and Helium from Flapjack. And again, these things are just also easier to show graphically. Whether this is actually the best graphic to do that or not remains to be seen. But this is just an example in terms of just various technologies that we've employed over time to make this work, in terms of, and one of the simplest ones just being flat files that we throw around in terms of either XML ones or plain text files. When you're just dealing with tools on the same desktop, is a really easy way to do that is going to be platform independent. You don't have to worry about firewalls and things. We have BRAPI, the plant breeding API, which I'll talk about in a moment, enabled for quite a few of our tools now in terms of how we integrate again between the tools and the data exchange, using things like the Galaxy API for tools, and then for integration of the data exchange, and then other simpler ways as well in terms of just basic web URLs that the desktop tools can call back to with some simple parameters to say, look, I have information. I want information on line X. Can you tell me that? And rather than duplicate the entire web interface for Germinate inside CurlyWurly or Flapjack, we can just make that call out to Germinate and say, look, can you display that information? And you can do that obviously with just really simple calls out to the browser and the URLs. So BRAPI, for those of you that are not familiar with it, I guess I should have had a quick slide on the overview of what it is, but it's the plant breeding API. It's a programmatic effort to bring together various databases and tools, both visualization and analysis tools and web-based systems that are developed, obviously not just by ourselves, but by people all around the world, particularly within the CG centers. So these tools can actually work together in a more seamless, simpler way for the end user. And that, if this tool X that does fantastic feature Y that you're interested in, but your data's in database running out of letters C or Z that you're interested in dealing with, you can pull them all together with BRAPI, and hopefully they'll all just talk to each other quite happily, and you can work from there. So in terms of what we've done with BRAPI and Flapjack and Germinate, then the genotypic visualization scenario or use case was one of the early ones that we actually had to deal with BRAPI. The genotypic data obviously adds up to quite a lot in terms of, if you're dealing with tens of thousands or millions of millions of markers, and obviously potentially tens of thousands of lines. And some of the early methods we had in there for dealing with that, when we tried to do this as a sort of individual HTTP call in terms of, okay, for this line, this marker, what's that allele called? And it just wasn't going to work at all. So lots of effort over time with the rest of the BRAPI group to just develop better ways, faster ways of dealing with that, and changes we've had to make over time as different databases and things have come on board as part of doing that. And as the sort of final point says there as well, what we've done, I don't know whether this is ironically or not in terms of doing that, is that while we have the BRAPI API to help us, we actually had to then develop another API to actually allow us to use the BRAPI API, just in terms of the Java code that we had to develop. But that, again, that API is there for anybody else that wants to actually then implement BRAPI in Java. We have that's potentially there for people to use. So there's a couple of slides here on some of the BRAPI calls. This doesn't really matter in terms of the technical detail. This was more just to sort of show that there were different routes of going through BRAPI to get genotypic information back out. But the key thing here is probably just to go back to Flapjack itself and show, just as a brief example, how that works. Where when we would go to the BRAPI import dialog, where normally we would be going to this maps and genotypes view and selecting the raw files that we're interested in, or if we hadn't pushed it from Germinate or from GOBI, then we have this ability to say, okay, we're interested in getting data from BRAPI. So a nice little just overview here that probably gives a better description of what I just did for what BRAPI actually is. Yeah, there we go. So it's an effort to create a RESTful specification to enable interoperability among plant breeding databases. So we say, okay, we're interested in bringing in data via BRAPI. We would then say, okay, so import via BRAPI. And then Flapjack at this point goes back to one of our servers and says, okay, well, which BRAPI data sources do we know about which are actually compatible with Flapjack? And it pulls back a few examples. So this is obviously one of our ones. We have a test Germinate dataset based off Cactar, which is a fictional crop from a video game. It's actually based on a fictional character from a video game that was shaped like a cactus, which is where we get cacti from. So it's a small demo data set. So we say this is what we're interested in selecting. For those of you that may be using this eventually with GOBii, and where the GOBii system is likely to be tied up behind institute firewalls and things, then obviously at this point you probably have to be providing custom URLs, which we can't host centrally that anybody that was using Flapjack is going to use. Anyway, so regardless of the way you actually get to the URL, you decide the data source that you're interested in getting. And we then say, okay, do you need to provide authentication details for that? Some of the options down here in terms of how it's actually going to break down and filter the data based on it, whether it's going to use study information, which is kind of like a way of wrapping up the concept of projects, whether it has map information to use or not. And this is sort of fairly reflective in terms of some of the challenges we've had in terms of developing BRAPI, and that each group and each database obviously all have their own terminology and nomenclature in terms of what they call and describe things as. You know, a study for one group might be an experiment for another, it could be a project for another group. So coming up with just a common language to make this work is still quite challenging. So regardless, you make your selections at this point. So in this particular case, Flapjack is going to filter the data by study, and it is going to attempt to pull back some map information. So the first thing it then says is, okay, well, which study you're interested in? Being demo data set, there's only one, or there would only have been one in this drop-down list here, and there's no details in it because it's just a demo data set. So it says, okay, there's a sample data set, that's what we're after. We then say, okay, well, which map are you after? So this is map number 1, which has all the markers. Again, you can see being demo data, it's full of pointless information in the database itself, but it does tell us that there's about 5,000 markers and 7 chromosomes. So I think this was data that was obviously probably generated from Barley. You do have the option even at this point that you could skip having a map, and it would just, Flapjack would make what we call a dummy chromosome instead, and all the markers are just equally spaced across that. So then the final thing is this concept of the actual data matrix that's going to be downloaded for the data and the number of lines that you're dealing with. So we've got, so just in the 5,000 markers, 2,000 lines. We don't know what type of data that is in terms of, you know, if you've got lots of heterozygotes, then that data set size could be bigger than if everything's homozygous. We don't know what the encoding is. But at this point, Flapjack tries to make a guess as to how big it will be. So obviously in this case, a 10 meg file, that's not too bad to try and bring that back. But, you know, if it said there was going to be a couple of gigs in size, you might want to bail out at this point and say, well, you know, there's just no point in attempting to use BRAPI to get data of that size. At which point it grabs it and it goes back to the standard Flapjack interface in terms of pulling back the lines and the markers and the alleles. And then eventually we actually get that data back into Flapjack where we can visualize it. Different color scheme here. I think this is, we have a color scheme based on allele frequencies, where if the frequency of particular alleles is low, it'll have one color, and if it's that percentage is high, then it gets a different color. One of the I'm not going to say easiest, but one of the easiest things we can probably do in terms of enhancing functionality in Flapjack for people is to add new color schemes to the tool itself. There are obviously some challenges around that, but if it turns out that, you know, if you have a particular type of data or data sets and the built-in color schemes that we have, and there must be about 10 or 12 default color schemes now, if one of them doesn't match what you need, then obviously get in touch and we can look at what the options are for building custom color schemes on top of that. And regardless of that, each of the color schemes that are there, I mean, you can tweak the colors for them as well. So I mean, it doesn't have to be green and blue in this particular case. So the last integration one I want to talk about briefly is just in terms of getting data from tools like Germinate and GOBii, and then bringing them back into Galaxy in terms of the nice ways that works. So this is the local Galaxy we have, where as you can see here, we've wrapped up a bunch of the analysis methods from Flapjack itself. But BRAPI is kind of a tricky way to pull data back into Galaxy because of the various API calls that you have to make, and because unfortunately the UI, the user interface in Galaxy is not great for the kind of decisions that you'd have to make. So, I mean, you can see even in Flapjack there, it wasn't a fantastic UI in terms of the wizard or the steps it went through to pick each section, and it would be even worse doing that kind of thing in Galaxy. So this is where Galaxy has quite a nice ability to deal with what it calls data sources, where you can say, okay, I want to import some information from Germinate. And when you click on that, what we actually do is we bounce out to a completely different website So we're no longer part of the Galaxy website here. We've actually gone, you can see the URL is different. But the key thing here is that there is this callback URL. It knows where it came from, that it came from this galaxy.hutton.ac.uk address. And at this point, because we have lots of different Germinate instances, we can say, well, which Germinate are we actually interested in pulling data from? And you would then select one of these at this point. And then you've got the really nicely designed Germinate interface to go through, visualize the data, work out what it is that you're actually interested in. And you just could not do this in Galaxy itself. You can't really do this in Flapjack either, even with the Brappy stuff, in terms of what data is it I'm interested in. Finally, make those decisions. And then you have this nice, simple option for the user at this point, just say send to Galaxy. And it's all automatic at this point because of that callback URL that was passed between the systems. Germinate knows how to talk to Galaxy and vice versa, and that information is then fed all the way back into Galaxy. And you can see here we have a sort of data matrix of genotypic information that can then be fed into some of the other Flapjack analysis. So the reason I'm sort of just covering all that in the integration scenarios is we, within the Hutton, have all these different tools. We know within the GOBI system there's all these different tools, and all the CG systems there's all these different tools. And the last thing I just want to mention briefly that we've been developing in collaboration with GOBI is this concept of a marker tools portal, which we're hoping to get out probably before the end of this year, I think is the current plan for it. And the idea here being just to bring together all of these tools into a single interface, while, or at least where you can launch them all, find out more information about them in one place, make sure that the installation that's required for the desktop tools is dealt with that way. Looking further forward, the idea is to be able to deal with the data at this point as well, that, if you have data in GOBI, then it would show up here, and then you can say, okay, we want to pass that to Flapjack or to KD Compute or something like that. So that's the longer term vision for it. But to begin with, it's just the idea of bringing the tools together, or all of the tools within the GOBI ecosystem will be in one place. So the ones that are based on the web, the ones that are based on the desktop. And one of the things we were discussing recently is, in terms of, unfortunately, we're going to end up with this hybrid scenario. We're going to have workflows where, for instance, if you're going to do a marker-assisted back-crossing analysis, you will start in the web in terms of extracting the data from whichever tool, making some other decisions on it, probably breaking things down by population, then launching it out to either Galaxy or to Flapjack, which is on the desktop. So the fact that at the moment that it says web or desktop is probably slightly misleading, because some of the, not so much the tools themselves, obviously, but the actual workflow will be this hybrid scenario. Okay, so another final slide on acknowledgements. So just to mention the team again. So Gordon, who is actually sitting here next to me and works very closely with me on both Flapjack and CurlyWurly and things like the Marker Tools portal and Tablet. David, who, as I was saying, used to be our group leader, he describes himself as a jobbing bioinformatician, which, whatever that means. But Sebastian and Paul that work on Germinate and Helium, and Linda, who works on some of the other web-based tools as well. So just to say thanks once again, thanks for all the collaborators and the funders that we've had over the years, because these tools would not have been possible without all of the help and the support that we've had. One of the things that we pride ourselves on is that we do continue to support these tools over many, many years, that we don't just knock out something over 6 months and then move, get a publication and move on to the next thing. We like the fact that we can develop and enhance and continue to support the tools that we have, but that would not be possible without the support that we've had from all of you. So thank you very much for that. So while this would normally be the slide that most people would finish on, I actually just want to jump to a final one, because it's probably a better one to just leave on the screen if we deal with any questions, which is to sort of summarize again. So that's where you can get the software. Most of the open source ones are there on the Crop Geeks GitHub as well, although one or two of them have slightly different what's the phrase to use, groups on GitHub, but they are still there, open source. And just to summarize again, so where they are desktop based, they all come with installers. There are no dependencies that you ever have to deal with yourself. They all automatically update, keep up to date. Whether web based, then obviously that's not a problem at all. And just as that final point again, that so we're always open to requests for new features, and we really want to help people where we can in terms of developing software. So I think that's me.
Thank you, Iain. It was a great overview of all the tools and the integration. So I'm going to open it up for questions from Any of the participants? Please just unmute your microphone if you want to ask a question, or you can also type it into the chat.
Hey, Gordon. This is Umesh.
Hi.
Can you hear me?
Go ahead, Umesh. We can hear you.
Yeah, I'm not used the tablet. You can make genotypic calls on it, or it's just a visualization tool?
Tablet's just visualization.
Yeah,
yes, you need to do your assembly or your alignments in other tools, whether that's things like Bowtie or Velvet, going back to some of the old school ones. But tablet itself is just the visualization side of it.
Yeah, and it would be great to have some tools like skim sequencing data where we can make genotypic calls and then export it to input as a blackjack. Then you have a connection of all tools.
Mm-hmm.
Just the idea, just floating.
Yeah.
Otherwise, your presentation is very nice. Very nice summary. Thank you.
Yeah, I mean, we can chat offline about options around that. I mean, I have no idea how much work would be involved in doing something like that. I mean, as I was saying, we spent an awful lot of time working on Tablet, and we really enjoyed doing Tablet at the time. But our focus more recently has been on the plant breeding side of things, which is why we've come back to tools like Flapjack. Yeah. Where I guess it's more of a niche space for us to fill with things like Flapjack, whereas there are other options for NGS visualization tools, which is slightly annoying for us because the longer we spend not developing Tablet, probably the further behind the rest of the tool space that has fallen. But that's just the way it works with, I mean, as I was saying, we have a team of bioinformaticians here of about 12 or 13, and you've seen there's only 4 or 5 doing software development, and even there, the 4 or 5 are not 100% software development.
Yeah, that's nice. I think it's a nice portal we have already, but it's just another idea floating. There can be several ideas, right? Thank you.
But yeah, ideas are always great because ideas are free.
Yeah,
that's the best bit. Yeah.
Yeah. But some ideas are more important than others, right?
That's the ones you then pay for.
Yeah.
Any other questions?
Hi, Ian. Yeah, this is Star. Thank you so much for the great overview. So really good to see so many different visualization softwares. So my question is, do you see the data management system currently, the EBS, has any adoption for any of the visualization software, the components you presented?
By that, do you mean the kind of diagrams and things that Tom Pagan has come up with in terms of how everything interlinks?
Right, with the before in the fundamental.
Yes, I mean, Flapjack certainly features in that, as does Germinate. I would assume CurlyWurly does as well, because I mean, that's heavily used by Summit. I mean, Tablet, like I say, is not really in the plant breeding space, so I wouldn't really see that fitting in there.
So Ian, I have a question for you about the haplotype space. I know that you've had some discussions, and so have we, about where and how we do haplotype analysis. To begin with, just like a local haplotype analysis. So where you're basically taking a region that's been selected and doing some kind of haplotype analysis on it and turning the visualization into a haplotype block. Which of your tools do you see this working in? Do you see that being a Flapjack application or one of your other tools?
Flapjack would be the most obvious one. It would all depend on what the requirements were for the tool itself. I mean, while I was saying, we're quite happy to support and extend existing tools, at the same time, we're equally happy to, start new tools if that's the best way to do things. Because again, it comes down to resource in terms of, the bigger and monolithic the tools get, the harder they are to then support and develop and extend. Sometimes it is easier just to spin up a whole new tool that's just focused on that one thing.
Mm-hmm.
And you could still have a situation where you make selections within Flapjack and it sends the data to that tool.
Right, I'm thinking about the haplotype tool that we're developing with IRI. There's an output from that I think we could feed directly into Flapjack. We'd have to look at all of the color matching issues, but
I think
then you get to multiple alleles, so I'm not quite sure how do you handle multiple alleles in Flapjack? Is that something you've looked at?
What do you mean? Do you mean beyond diploid?
Yeah, well, when instead of having 2 SNP alleles, you might have up to 10 different haplotype alleles.
Yeah, I mean, Flapjack copes with it behind the scenes in the data model, but it's one of these things we've just shied away from trying to work out the best way to visualize it, because until fairly recently, there really wasn't that need for it. And all the people that were dealing with those kind of crops just weren't interested in using the tool. So we're focused on the diploid side of things. There will be a way. I mean, anything's possible at the end of the day. So one of us or somebody will come up with a nice way of, whether it's visualization or something else, of coping with that. But I couldn't give you an easy answer just now.
Mm-hmm.
Yeah, to the point, Miss, again, that we're developing the mass tool in Flapjack that STPG supported. So if you have haplotyping, that will help to that tool too, that we already discussed about.
Right.
Yeah.
Lots to think about. And Iain, another question about, I know you work a lot with the gene bank data, which is bulked samples and then storing that data in Germinate. How do you handle the kind of data that they have, where you might have frequency data for a particular haplotype?
I can't answer that because it's not me that deals with that directly.
Okay.
Looking at Gordon, but I mean, I don't know whether you're anymore.
No, I think you probably have to.
Sebastian's probably the best person. We can get you an answer. I mean, there is some allele frequency stuff that's in Germinate. We actually have some visualizations around Flapjack for that as well, but I don't know if that's the same thing that you're talking about here. I'm going to go back on my get out of jail free card from the start in terms of that my background is computer science, not biology.
Yeah, that would be good to follow up on, because that's something that Sarah wanted us to do a while back, and we haven't really followed up on it, how to handle that kind of data. And it'd be good to align with what you're doing in Germinate.
I mean, if you fling something to us in an email and we'll get you an answer.
Okay, will do.
Hi, Iain. So for CurlyWurly, is that you need someone to really set up Yonder for broader users to use the tool to select?
I'm not quite understanding the question there, Star. So CurlyWurly is a desktop-based tool.
Right, yeah. So once you have the data in CurlyWurly, right, you have a lot of pedigree information. Is that in a group, one person need to really set up this with the size, the color tune, or just any user can use directly from, like, say, Excel data output to the visualization?
Yeah, I think it's something that any user can use to explore their data, and be able to look at the different categories and make their selections and sizing of the points within the view based on what we're seeing. It's a very interactive tool. It's quite easy to use. So the best thing to do is sort of dig in and just play with your data.
And what are the inputs, Gordon? Is that an allele matrix, or do you have to do the PCA analysis before going into CurlyWurly?
You do the PCA analysis before going into CurlyWurly. The data format is explained on our documentation website. One discussion we have been having internally is whether we try and alter the way that CurlyWurly works just now, and have it, allow you to provide it with an allele matrix, and then you could have different distance measures that you use, depending on what you're wanting to do with the data, and potentially different types of clustering before you actually get to the 3D view.
So it's a workflow that it's a little separated right now, but you're thinking about integrating it more.
Yeah, and the question is about whether you just sort of turn CurlyWurly into a sort of adjunct to Flapjack, or if you put more features into CurlyWurly.
Right. Okay, so we're coming up to the end of the hour. So thank you very much, Iain and Gordon, for answering the questions at the end there.
No problem. Yeah, no
really
problem.
Great presentation, and I believe Star will be posting the recording and the slides on YouTube, and she'll send out a link.
Okay, I'll send you an email with the presentation as well, Star.
Sure. Okay. Thank you very much. Yeah.
Thank you, everyone. Bye-bye.
Okay. Have a good day now. Thank you so much. Bye-bye.